Hello friends, welcome to my channel. Today we are going to study here the 12th standard biology. In this video, we'll study the chapter number 1, that is reproduction in lower and higher plants of 12th standard biology. In previous video, we had studied asexual reproduction, budding, binary fission, gemule formation, cutting, tissue culture, fragmentation, pore formation, conidia formation, vegetative propagation, and grafting. We have already studied all these points in our previous video, and now in this video, we will cover the following points: sexual reproduction in plants, flower, the structure of flower, sexual reproduction and its events, characteristics of sexual reproduction in plants, the structure of anther, TS of anther, microsporogenesis, the structure of microspore, development of male gametophyte, structure of anther and the structure of anatropous ovule, megasporogenesis, and the development of female gametocyte. In this video, we will study all these points. Sexual reproduction in plants. In general sense, sexual reproduction in plants, particularly in flowering plants called angiosperm, is a process by which new individuals are created through the fusion of specialized reproductive cell called gametes. This process involves two parent plants contributing genetic material leading to offsprings that are genetically diverse from both parents. So, as you can see in this image, the whole process of sexual reproduction in plants is showing over here. So, here we see the definition and the process of sexual reproduction in a plant. Okay, so the definition is more specific and it involves a series of sequential events centered on the flower, which is the reproductive organ. Here the flower is a reproductive organ in the plant. Okay, the key components of this process are as follows. The first step of this process is the formation of gametes. This process is also called as the gametogenesis. The certain of male the creation of male gametes. The male gametes is also called as the sperm within pollen grains, and the female gametes, which is also called as egg cells within the embryo sac of the ovules. The second one, pollination. The transfer of pollens from the anther. Anther is also called as the male part to the stigma. Stigma is also called as the female part of a flower. The third step is fertilization. This is the main step. Okay. The fusion of male gametes within the egg cell, forming a diploid zygote. This is followed by a unique event in the flowering plant called as the double fertilization, where the second male gametes fuses with the polar nuclei to form a triploid endosperm, which will nourish the developing embryo. Now, after the fertilization, some events will take place called as the post-fertilization events. Okay, after the fertilization, post-fertilization. The development of zygote into the embryo will take place here. The ovule into a seed and the ovary into a fruit. This process is crucial for introducing genetic variation, which helps a species adapt to changing environmental conditions. Now, here we see the overall re- review about the flower. Okay, flower is defined as the reproductive unit of an organism. For the flowering plant, okay, here we are talking about the flowering plant. Okay, it is highly modified shoot or stem whose primary biological function is to carry out sexual reproduction by facilitating the fusion of male and female gametes, leading to the formation of seeds and fruit. Okay, this is a definition of flower. Now, the structure of a typical flower is composed of four main whorl. Whorl means parts arranged on a swollen end of a flower stalk called the receptacle. So what is mean by receptacle? The end of flower stalk, okay, which is swollen. These whorls are calyx, that is non-essential whorl, corolla, that is also a non-essential whorl. The next one is androecium, essential whorl, and it is a male reproductive part as you can see in the image. The next one is gynoecium or pistil, that is also a very essential whorl and that is also called as the female reproductive part. Now the gynoecium contains the three different parts: stigma, style, and ovary. The structure of a typical flower. So here we'll see a flower is composed of four main whorl. Whorl means parts arranged on a swollen end of a flower stalk called as receptacle. These whorls are, the first one is calyx, which is non-essential whorl. So what is the structure of calyx? It is the outermost whorl of the flower consisting of green or leaf-like structure called as sepals, as you can see in the image. So, and what is the function of this part? It is its main function is to protect the flower in its bud stage from mechanical injury, dislocation, or drying out, and herbivorous. The second one is corolla. It is also a non-essential whorl. The structure of this part is the whorl located just inside the calyx, made up of brightly colored petals. Now, what is the function? The vibrant colors and fragrances of petals serve to attract pollinators like insect, birds, and animals, which are essential for pollination. Now, the third one is an androecium, essential whorl, and also called as the male reproductive part. Now, what is the structure of androecium? This whorl is made up of stamen. Each stamen consists of a long cylinder stalk called the filament and the bilobed head called anther. And what is the function of anther? The anther contains pollen sacs where pollen grains, also called as the male gametophytes, are produced. The filament holds the anther in a position to facilitate pollen dispersal either by wind or by pollinators. Now, the last one is gynoecium or pistil. It is also a very essential whorl. It is also called as the female reproductive part. Now we'll see the structure of gynoecium. This is the innermost whorl composed of one or more carpels. The carpels has three parts: stigma, style and ovary. Now we'll see the overview of stigma. It is sticky, receptive tip that it traps the pollen grains. The next one is style, a cylinder stalk which is connecting the stigma to the ovary. It is basically it is a tube-like structure. Ovaries, a swollen basal part that contains one or more ovules. Basically, the structure of ovary is a sac-like structure. Now, what is the function? The gynoecium is where the female gametes, what is the female gametes? Egg cells are formed within the ovules. It is the site of fertilization, after which the ovary develops into a fruit and the ovules into seeds. Sexual reproduction in plants and its events. Now here we'll see what are the events takes place during the sexual reproduction process in a plants, especially in a flowering plant. Okay. The study of sexual reproduction in plants is centered on angiosperms. Angiosperms means the flowering plants, but also it includes a broader understanding of the plant kingdom. The major events of sexual reproduction in flowering plants are as follows. Okay, the process of sexual reproduction in flowering plants can be divided into three major or sequential events. The first one is the pre fertilization event. In pre fertilization events, there are mainly three parts. The first one is the gametogenesis, pollination, and pollen pistil interaction. So, here we'll see the overview of all these three steps. Okay, the first one is the gametogenesis, the formation of haploid male and female gametes. This includes microsporogenesis, means the formation of pollen grains in the anther, and megasporogenesis, means the formation of embryo sac containing the egg cells in the ovule. The next one is the pollination. Pollination means the transfer of pollen grains from the anther to the stigma, which can be achieved by agents like wind, water, or animals. Now, the next is the pollen pistil interaction. The recognition of the correct pollen grains by the pistil, leading to the growth of a pollen tube down the style to the ovule. Now, the next step is the double fertilization. Okay, so students don't get confused. In pre fertilization events, three steps are there: gametogenesis, pollination, and pollen pistil interaction. Okay, now the next one is the double fertilization. This is a unique and defining characteristics of angiosperms. The pollen tube releases two male gametes into an embryo sac. Now here we see the C1 point, that is syngamy. Syngamy is also called as the true fertilization. One male gamete fuses with the egg cell to form a diploid zygote, which is two in number, which will develop into an embryo. Now, the triple fusion. The second male gamete fuses with the two polar nuclei in the central cell, forming a triploid primary endosperm nucleus. Okay, it is also called as the PEN, which will 3 in number. This will develop into a endosperm, a nutritive tissue. Okay, the last one is the post fertilization events, which will takes place after the fertilization. Okay, and it includes endosperm and embryo development. The PEN divides to form the endosperm, while the zygote develops into the embryo. Seed and fruit formation. The last step. The ovule matures into the seed with the integuments of the ovule becoming the protective seed coat. Simultaneously, the ovary develops into the fruit, protecting the seed and aiding in its disposal. Characteristics of sexual reproduction in plants. The first one is the alternation of generations. The plant life cycle involves an alternation between a diploid sporophyte generation, means which produces spore by meiosis, and a haploid gamete generation, which produces gametes by mitosis. Now, the next one is the genetic variation. The fusion of gametes from two different parent, means the cross pollination, results in offspring with new genetic combinations, which is crucial for adaptation and evolution. Next one is a double fertilization. The simultaneous fusion of one male gamete with the egg cell and the another with the central cell is a distinctive feature of flowering plant. Now, the last one is the formation of a seed and a fruit. The entire process is contained within the flower and culminates in the formation of a protected seed within a fruit. Three group of sexual reproduction. Alternation of generations. This concept of three groups most commonly refer to the three distinct patterns of alteration of generations observed across the plant kingdom. This is crucial topic for understanding plant evolution. The first will see the haplontic life cycle. So it describes the dominant or a free living plant body is a haploid gametophyte. Okay. The diploid sporophyte, which is 2 in number, is only represented by the single celled zygote, which undergoes meiosis immediately after fertilization. Examples: many algae like Spirogyra. The next one is diplontic life cycle, which describes the dominant or a free living plant body in a diploid sporophyte, which is 2 in number. The gametophyte generates is highly reduced and is represented by only a few cells. Example: pollen grains and the embryo sac. Okay, so the examples are all seed bearing plants, angiosperm and the gymnosperm. Now the next one is haplodiplontic or haplodiplontic life cycle. It describes It describes both the haploid gametophytes and the diploid sporophytes are multicellular and distinct phases in the life cycle. The degree of dominance varies between different plant groups. So here are the examples. Bryophytes. Example, mooses. The dominant phase is the haploid gametophyte. The sporophyte is dependent on the gametophyte for nutrition. The next one is pteridophytes. Example, ferns. The dominant phase is the diploid sporophyte, but the gametophyte is also a free living independent plant body. Androecium. What is mean by androecium? The male reproductive whorl of a flower is called as the androecium. This whorl is composed of individual unit called stamen. As you can see in a figure, each stamen is a male reproductive organ and it is made up of two main parts, a filament and an anther. Here is detailed breakdown of a structure and function of each component. The first one is the androecium. Okay, so the definition of androecium is the term androecium refers to the entire collection of stamen in a flower. It is the third whorl of the floral arrangement located inside the corolla, and the function of androecium is its collective function is to produce and release pollen grains which contain the male gametes necessary for fertilization. The second thing is the stamen. Now, what is mean by stamen? The stamen is an individual unit of androecium. It represents a single male reproductive organ. The function of stamen is the primary function of stamen is to produce the pollens. It is composed of a filament and a connective and an anther. The third one is filament. The filament is a long, cylinder, stalk like support that anther. Its proximal end, the base, is attached to the thalamus receptacle or in some case to the petal of the flower. The function of filament is the filament's main role is to hold the anther in the position that facilitates pollen dispersal either by wind, water, or a pollinator. It is also served as a vascular connection, supplying nutrients and water to the anther. The fourth one is the connective. The connective is a sterile tissue that connects the two lobes of the anther. It is essentially a continuation of the filament. The function of connective is it holds the two anther lobes together and contains a vascular stand that runs from the filament into the anther. Now finally it comes the anther. Anther is a terminal typical bilobed structure at the tip of stamen. It is a fertile part of a stamen where pollen is produced. Now we'll see the structure. A typical anther is a four-sided tetragonal structure with four chambers called microsporangia or a pollen sac with two in each lobe. Each microsporangium is where microsporogenesis occurs, leading to the formation of pollen grains. Now, the function of anther. The primary function of anther is to produce, store, and release the pollen grains. When the anther matures, it undergoes a process called dehiscence, where it splits open to release the pollens. The structure of anther. A typical anther is a bilobed, means it contains two lobes and dithecus. Each lobe has two chambers or trays. Each lobe has two chamber or thica thica structure. It is also considered tetrasporangiate because each of its two lobes contains two microsporangia, pollen sacs, for a total of four. These pollen sacs are where pollen grains are produced. The anther is supported by a stalk like structure called the filament anther wall layers. The anther will consist of four distinct layers from the outermost to the innermost. The next is epidermis. It is the outermost single layer of cell. Its primary function is to protect. The next is endothecium. It is located just beneath the epidermis. This layer is made up of radially elongated cells that develops fibrous thickening of a cellulose. These fibrous bands are hygroscopic. They absorb moisture, which cause the anther to split open at maturity, releasing the pollen grains. The next is the middle layer of anther. These are typically 1 to 3 layers of cell that lies between the endothecium and the tapetum. They are generally short lived and get crushed as an anther matures. They store such and provide nourishment. The next is tapetum. It is the innermost layer of the anther wall. It surrounds the sporogeous tissues. The tapetum is crucial for nourishing the developing microspores called the pollen grains. Its cells have dense cytoplasm and are often multinucleated. This layer also secretes enzymes and hormones required for pollen development, including the enzyme cellulase, which dissolves the la- layer, cellulose layer that holds the microspore together in a tetrad. The next one is microsporogenesis, also called as the pollen formation. Inside each microsporangium, there is a mass of homogeneous cells called sporogenous tissues. The cells of tissue act as a microspore or mother cell or pollen mother cell. Each microspore mother cell undergoes meiotic division to produce a cluster of four haploid cells known as a microspore tetrad. As in another, as the anther matures, the microspores separates from the tetrad and develop into pollen grains, which contains the male gametes. In typical angiosperm, the anther is a male reproductive part of the flower where pollen is produced. A transverse section, that is TS, of a mature anther reveals a highly organized internal structure with distinct layer and tissues. Now we'll see here the anther structure in TS. A typical anther is a bilobed, means it has the two lobes, and diathys structure, meaning it has two lobes and each lobe contains two pollen sac or microsporangia. It makes the anther tetrasporangia, four sporangias. The two lobes are connected by a sterile tissue called the connective anther wall layers. Okay, the transverse section of a young anther shows four layers arranged connectively around the central sporangious tissues. The first one layer is the epidermis. This is the outermost single layer of cell. Its primary function is to provide protection to the anther. The second layer is the endothecium. It is located just beneath the epidermis. This layer consists of rapidly elongated cells with fibrous thickenings of a cellulose. These fibrous bands are hygroscopic. Hydroscopic means water absorbing, which is essential for the dehiscence or splitting open of the anther to release pollen when it is mature. The third one is a middle layer. These are typically three layers of cell found between the endothecium and the tapetum. tapetum. They are generally short lived and get crushed as anther matures. They serve as a temporary storage and transport layer of nutrients to the inner tissue. The fourth one is tapetum. It is the innermost layer of the anther wall. Its cells are rich in cytoplasm and are often multi multinucleates, developing pollen grains called microspores by providing essential nutrients, enzymes, and hormones. It also helps in the formation of sporopollens, pollenin, sporopollenin, a resistant substance that makes up the outer wall of the pollen grain. Now here after that we'll see here sporogenous tissues. It is located at the center of each microsporangium. The sporogenous tissues is a compact mass of cell. The cell of this tissue undergo meiotic division to form microspore tetrad, tetrads, which eventually mature into individual pollen grains. Now here we'll see about the microspore. In flowering plant, a microspore is haploid N spore produced from a diploid microspore called the mother cell or pollen mother cell through meiotic division. It is the first cell of male gametophyte. The process of its formation is called microsporogenesis and occur within the microsporangia called the pollen sac of the anther. After meiosis, the four microspores are initially arranged in a cluster called a microspore tetrad. An enzyme called cellulase secreted by the tapetum layer of the anther wall dissolves the cellulose layer that holds the tetrad together, releasing the individual microspores. Now here we'll see the structure of microspore. As you can see in an image, the microspore which later matures into a pollen grain is typically a single spherical cell with a prominent nucleus and dense cytoplasm. It develops a two-layered wall. The first one is exine. This is a hard outer layer of microspore. It is composed of sporopollenin, one of the most resistant organic substances known. The presence of sporopollenin makes the exine highly resistant to the high temperatures, strong acids, and alkali, protecting the pollen grain from degradation. The exine is often sculptured with unique patterns that are characteristic of the plant species. At certain points, the exine is thin or absent, forming structure called germ pore or germinal furrows. These are the sites from which the pollen tube will emerge during germination. The second one is intine. This is the thin inner layer of microspore wall. It is composed of cellulose and pectin. The intine is elastic and continuous and it surrounds the cytoplasm and nucleus of the cell. After the microspore is released from the tetrad, it undergoes unequal mitotic division to form two distinct cells. The next one is vegetative cell, a larger cell with abundant food reserves and an irregularly shaped nucleus. It is responsible for forming the pollen tube during germination. The next one is generative cell, a smaller cell with dense cytoplasm and a prominent nucleus. It is initially attached to the wall of the vegetative cell and will never divide to form the two male gametes or sperms. Now we'll see here the development of male gametophyte. The development of male gametophyte in flowering plant, also known as microgametogenesis, is a key part of the plant reproductive cycle. It begins with a microspore and culminates in a mature pollen grain containing male gametes. Here is a step by step breakdown of this process. The first step is microspore stage. In this process, the process starts with a single haploid microspore which is the first cell of a male gametophyte. The spore is surrounded by a two-layered wall. The tough outer layer called the exine, made up of resistant substance called sporopollenin, and the thin layer called the intine, made of cellulose and pectin. The second one is first mitotic division. In this process, the nucleus of the microspore undergoes an asymmetrical mitotic division. This division results into two unequal cells. A large, irregularly shaped vegetative cell, also known as the tube cell, with abundant food reserves and a large nucleus. This cell is responsible for forming the pollen tube after pollination. A smaller, spindle shaped generative cell that is initially attached to the microspore wall but soon detaches and floats in a cytoplasm of the vegetative cell. The third one is a pollen shedding. In about 60% of angiosperms, the pollen grain is shed from the anther as this two-celled stage, one vegetative cell and the one generative cell. In the remaining 40% of plant like sunflower, the generative cell divides again inside the anther and the pollen is shed at the three-celled stage. The fourth one is second mitotic division, also called as post-pollination. After the pollen grain is shed and lands on the stigma of a flower, also called as pollination, the vegetative cell absorbs moisture and nutrients from the stigma. The intine, the intine grows out through one of a germ pores in the exine, forming a pollen tube. The generative cell moves into the pollen tube. The generative cell then undergoes a second mitotic division to produce two non-motile male gametes, also called as the sperm cells. The fifth one is mature male gametophyte. The mature male gametophyte is the three-celled structure, one vegetative cell and the two male gametes enclosed within a pollen tube. This pollen tube grows down through the style and into the ovule, delivering the two male gametes for fertilization. Anatropous ovule. The anatropous ovule is the most common type of ovule found in flowering plant called angiosperms, characterized by its inverted or bent structure. This unique orientation is an ov- evol- evolutionary adaptation that helps to ensure efficient fertilization. Now we'll see the structure of anatropous ovule. The ovule is a small ovule structure attached to the ovary wall by stalk. It is a key its key parts are funiculus or funicle. This is a stalk that attaches the main body of ovule to the placenta inside the ovary. The second one is hilum, the point where the funiculus connects to the body of the ovule. The third one is raphe, the fused portion of a funiculus with the ovule's body, which appears as a ridge. In an anatropous ovule, due to the 180-degree inversion, the funiculus lies fused with the body of the ovule. The next one is integuments. These are one or two protective layers that surrounds the ovule. They are protective envelopes of a nucleus and will later develop into the seed coat after fertilization. The next one is micropyle, a small opening or pore at the apex of a ovule. It is the only area not covered by the integuments. In an anatropous ovule, the micropyle is located close to the funiculus. The next is nucellus. This is the main body of the ovule, a mass of cells that is enclosed by the integuments. It provides nutrition to the developing embryo sac. The nucleus is equivalent to the microsporangium. The next one is chalaza. This is the basal part of the ovule where the integuments and the nucleus originate. It is located directly opposite to the micropyle. Next, embryo sac or female gametophyte located within the nucleus. The embryo sac contains the egg cell and another associated cells. It is where fertilization takes place. Now we'll see the function of an anatropous ovule. The primary function of the ovule is to participate. The primary function of the ovule is to participate in sexual reproduction by facilitating fertilization and developing into a seeds. The specific structure of the anatropous ovule aids this process in several ways. Like the first one is efficient fertilization. The inverted orientation brings the micropyle close to the funiculus, allowing the pollen tube to enter the ovule more directly and easily after traveling down the style. The next one is protection. The integuments serve as a protective layer, shielding the delicate nucleus and embryo sac from mechanical damage and desiccation. After fertilization, they harden to form the tough seed coat. The next is nourishment. The funiculus provides a pathway for transport of nutrients from the parent plant to the developing embryo sac and later to the embryo inside the seeds. The nucleus also serves as a source of food. Micro megasporogenesis. Megasporogenesis is a process of formation of haploid megaspores from a diploid megaspore mother cell. This crucial event takes place inside the ovule of a flowering plant. The process can be broken down into the following steps. The first one is megaspore mother cell. Within the nucleus, the main body of a ovule, a single diploid cell differentiates and becomes the megaspore mother cell. This cell is larger than the surrounding cell, has a dense cytoplasm, and a prominent nucleus. The second one is meiosis. The diploid mother cell undergoes meiotic division, meiosis first and meiosis second, to produce four haploid megaspore. This process is reductional division, meaning the chromosome number is halved. The third one is linear tetrad formation. The four haploid megaspores are typically arranged in a single file, forming a linear tetrad. The fourth one is degeneration of megaspores. In the majority of flowering plants, over 70%, a type of development called monosporic. Only one of the four megaspores remains functional. The another three, usually located towards the micropylar, and generate degenerated. This single functional microspore is typically the one located at the chasal end. This functional megaspore will then undergo further divisions to develop into the female gametophyte or embryo sac. The development of female gametophytes. The development of female gametophyte is also known as the embryo sac, is a two-phase process. The first phase is the megasporogenesis and the second is megametogenesis. Now we'll see the first one, megasporogenesis. This is the first phase where a diploid megaspore mother cell located in the nucleus of the ovule undergoes meiotic division. This division produces four haploid megaspores in a linear tetrad. In most flowering plants, known as monosporic development or tetrad. In most flowering plants, known as monosporic development or polygonum type. Only one of these megaspores, usually the one at the chasal end, remains functional. The other degenerates. Now we'll see the second phase, that is megagametogenesis. In this phase, the single functional megaspore develops into the mature female gametophyte, that is embryo sac. The nucleus of the functional megaspore undergoes three successive mitotic divisions. These divisions are free nuclear, meaning that the nucleus divides without the immediate formation of cell walls, resulting in a single cell with 8 haploid nuclei. The mature embryo sac is a seven-celled, 8 nucleate structure organized as follows. The first one is egg apparatus. It is located at the micropylar end. This consists of three cells. One egg cell, this is the female gamete that will fuse with a male gamete during fertilization. And the second one is the two synergid cells. These cells guide the pollen tube towards the egg cell with the help of a specialized structure called the filiform apparatus. Now, the next one is the central cell, the largest cell in the embryo sac located in the center. It contains two polar nuclei which will fuse with the second male gamete during double fertilization to form the triploid primary endosperm nuclei. Now, the next one is antipodal cells. Three cells located at the chasal end of the embryo sac. They typically degenerate after fertilization. This entire process from the single functional megaspore to the mature seven-celled and eight nucleate embryo sac is crucial for the process of double fertilization, a unique characteristics of angiosperms. I hope you like this video. Thanks for watching. Follow us on Instagram and YouTube Shorts. Like, share, and subscribe. And click on the bell icon to get notified about every new video.